evening. Welcome back. Welcome to my channel, Zoela Bomba. We are listening to Divertimento by Moses, and it is the title for the chapter I presented part of uh, my last entrance on this channel, and I talked about Divertimento chapter 6 of Richard J. Miller, the author of Drug, the Science and Culture Behind Psychotropic Drugs. Uh, in the first the first part of this chapter, I talked about war, about World War One, World War Two, how people were trying to seek the entertainment and relax in the times of wars, where everybody is having a doom hovering over his or her head. And the chapter opened on a theater for music, and there was a divertimento piece being presented that time, and then that's how he introduced the disorder of anxiety, the and all the ailments that come with war. We've talked about also about certain medications that treat this disorder, among others, but initially they were not sought to treat this particular disorder. Uh, Clinically, it's very recent as an established disorder in the field of psychiatry. Since psychiatry has started to follow a new treatment line, which includes pharmacology. So in the chapter we talked about mefenicine, about meprobamate, about equinil, which is the same as melatonin. And then we talked about benzodiazepines and how all these medications and barbiturates and about all these medications, they I love to say stumble because there is lots of hazard in it. There is lots of chances in it. It was not something that they knew we're gonna put this formula together with this formula and we're gonna come up with this. They, they were not even looking for medication for that very particular disorder. That's why the author said that the serendipity was involved in addition to excellent science. So mefenicine came as they were looking for a preservative for penicillin, and then they, it came with lots of side effects and narrow therapeutic index, and then short acting. So it was inefficient. It was had the lethality side effect, and then they further searched it and came out with meprobamate, and this medication proved to be muscle relaxant more than anything, and also they proved to not relax the patient. They tried them on rodents first without putting them to sleep. So, and then they used them for epilepsy, for MS, for and as muscle relaxant, as sedatives, as pre-anesthetics and as anesthetics, and then after meprobamate came mefenicine, came Milton, which also which is also traded as sold as equinil. Then they wanted to look for all these have a narrow therapeutic index, so they wanted to come up with something better, and then they came up with only barbiturates. Barbiturates was good for as a hypnotic, as anesthetic, as a sedative, and also possibly was used for epilepsy also. But then it also has a narrow therapeutic index. They came up with an antidote for it. they make great, and which is a stimulant. And then the problem with the barbiturates, it was addictive, and also in addition to its narrow therapeutic index, and it was used also in as a in, in street drugs, mixing it with amphetamine, and then making it like what they call goofballs. And there was the overdose crisis among people spiked, and there were lots of deaths and all that. And they started to search, although barbiturates are still used though, but they started to use for different things with less lethality side effects and more effective or efficient as muscle relaxants, as sedatives, as hypnotics, as anesthetics, and uh, and each with their therapeutic profile or their own utility, and that's how they came to benzodiazepine and how Lewis Sternbach was involved in this. If you wanna see all these facts in detail, you go back to the previous post and then have a look at it. And then if you have any doubts and you want to verify, go to the book itself and read it or listen to it, and then do your own research. But for me, it's just a way to share what I know and what I wanted to know more about. And the book has a way of presenting these things. They start with the historics, and then how these medicines came to the world and who was involved in bringing them to us, and then why they work the way they work, and what do they work on. And that's with a very actually premeditated uh, uh, thinking in mind, which says that since all these medications, they have receptors in our brain or in our bodies, then why do our bodies inside have those receptors in? And then we end up finding that there are similar chemical substances or substances with similar uh, chemical um, structures in our bodies, just how they work, and that's what he wants us to understand. Fixing these things probably it is something that needs further research, and I'm not an expert in the field. I am with a person with some basic knowledge in chemistry, and I just love to read about it. I had nursing education, and I had some scientific education here in US, and I also I'm a certified EMT, and I'm certified in applied health. And previous to that, I was a teacher of English, which is way different from anything. I was also a writer, but I love science. I love to profound my knowledge in scientific field, and I love to be able to, you know, be actually somebody who's practicing in the field with more uh, in-depth knowledge about the field with hand-on experience, not only theoretical experience. So, leaving this aside, let's go on and talk about what's coming in the second part of what I present as the second part uh, in this uh, in this episode. Shall we call it? So, so now let's talk about the benzodiazepine and receptors. Scientists at Roche worked relentlessly to decipher the mechanism of action of benzodiazepines in 1970. Similar to what we have seen with what Snyder Solomon did to find opiate receptors, they used bioassays or receptors binding assays. They irradiated or tritiated, they used tritium. They irradiated diazepam and used it on animals and observed. It was noted that some benzodiazepines are stereoisomers, similar to what has been found about opiates, as mirror images of themselves. One isomer is an active benzodiazepine and the other is not. And we've seen that with morphine, right? The binding site has high affinity for tritium labeled active benzodiazepines, but not so much for the inactive one. Autoradiography also used and allowed for exact visualization of the binding site structures in the brain, widely diffuse and markedly heterogeneous, abundant in some areas and scarce in others, identified as protein molecules. The question that arises again, what is the actual physiology of these sites that obviously existed in the brain before benzodiazepines? These sites were in the brain before we discovered benzodiazepines. So, what was their actual utility? Now, are there occurring benzo-like molecules the human or animal body naturally synthesize? The answer lies in the electrophysiology, as the author says, measuring the electrical activity and action potential within the cells and neurons, as the author will explain later. Now let's see the GABA connection. The GABA is, if we break it down, gamma aminobutyric acid. So the GABA connections. Experiments run by neuroscientists demonstrate that diazepam or benzos or benzodiazepines in general potentiate the inhibitory effect of GABA neurotransmitters. Benzos, as a slang for benzodiazepines, do not influence GABA metabolism, but rather affects its synaptic receptors. They're just modulated. They do not act directly on it. As we have seen in chapter 1, there are two types of GABA receptors, GABA-A receptors and GABA-B receptors. GABA-A receptors are ligand-gated ion channels, while GABA-B receptors are GPCRs, proteins coupled receptors, or G protein, guanine, nucleotide binding proteins, largest signaling receptors in the cell, plasma membrane. Benzodiazepines action could be blocked by drugs that block GABA-A receptors, such as picrotoxin. Benzodiazepine and neurotransmitter GABA both bind the GABA-A receptors, which has a complex size. Both GABA and benzodiazepines are small molecules and bind to different sites of GABA-A receptors without overlapping. Benzodiazepines and the neurotransmitters GABA both bind to GABA-A receptors, which has a complex size. Both GABA and benzodiazepines are small molecules and bind to different sites of GABA-A receptors without overlaying. So GABA-A receptors are large molecules, while GABA, which is neurotransmitters, and benzodiazepines are small molecules, and they bind to that very receptors without overlapping. They have their own, each has its own site. But benzodiazepine facilitates the work of GABA. Benzodiazepine do not directly influence the site, as I told you, but facilitate the GABA's tasks through what is known as allosteric mechanism, where the site is a lock. GABA is the key benzos. The benzos act as the greasing oil, like they grease the key for GABA to get in. GABA-A receptors are abundant in the brain. Every neuron has a receptor, and GABA is a major neurotransmitter. Benzodiazepines affect the body in many ways. They can act as anxiolytics, sedatives, hypnotics, anticonvulsant, muscle relaxant, and can have a retrograde amnesia effect. GABA-A receptors are heterogeneous. That means they are abundant in certain area and scarce in another, and made of different protein subunits. There are also different GABA-A subtypes. These GABA-A receptors have also different subtypes. Do benzodiazepine binds to every GABA-A subtype receptors, or are they selective, and do they affect those subclasses differently? You have subtypes or subclasses of GABA-A receptors, and we know benzodiazepines bind to these. And do they bind to every subclass, or do they have different effects on each class of these receptors? If benzodiazepines are similar in effect to drugs that affect a certain subclass and produces selective effects, if so, this would be clinically significant. Long-term use of benzodiazepines, however, results in serious dependency. If the anxiolytics and addictive effects are separated, it results from let's say the anxiolytic and addictive effect results from their interaction with different subclasses or separate subclass GABA-A receptors. This could change. It's like it's a very important fact, because that it could change it all. They could separate the effect in the medication, and then we wouldn't have that addictive effect. They would like work on the medication itself, and then know exactly which one affects which that very particular receptor that causes the addictive effect, and work on it. Molecular biological studies prove that GABA-A binds to a receptor at the sites between alpha and beta subunits, while benzodiazepines bind to sites between A and gamma-2 subunits. The facilitator role benzos play happens only on subgroup A receptors that contain gamma-2 subunits. See, now we're getting clearer idea. Benzos bind only to those containing gamma-2 subunits and certain alpha subunits. Molecular science studies at Hoffman-LaRouche tries then to develop new, better drugs that could bind to GABA-A receptor subtype benzodiazepines bind to for specific effects. Experiments on genetically modified mice showed that drugs that selectively bind to GABA-A receptors are hypnotic sedatives without negative side effect, the anxiolytic side effects. I mean, they can be hypnotics, but do not affect anxiety. Yeah. So the author said those studies were conclusive, but also said the drug might have sedative effects without the anxiolytic effects, which is not as certain. Using might does not imply that kind of certainty that the word conclusive does. Ambien, zolpidem exhibits such selectivity as a hypnotic agent. Thousands of drugs have been since then that interacts with the receptor sites with which benzodiazepines interacts with a plethora of different effects, from hypnotizing to sedating to anesthetizing to death occurrence. Some of them can cause death. All these drugs are and share certain effects but produce specific effects by binding to GABA-A receptors in a unique way. Now let's relax and listen to some Divertimento. This is an anxiolytic without the addictive effects. Okay, now let's see and contemplate the brain on steroids. It has been known that there is actually a biomolecule that naturally interacts with GABA-A receptors. Uh, it's GABA, the, the natural uh, substance that's produced by the body and interacts with the GABA-A receptors is GABA. So in 1927, it was observed that cholesterol plays an inhibitory role in the brain. So it's been observed that cholesterol acts the same way GABA acts. It plays an inhibitory role in the brain. Cholesterol is important for cell membrane synthesis, cell signaling, and also important for steroid hormones synthesis. It was demonstrated in 1940s that synthetic steroids derived from cholesterol produce sedation. So alfaxalone, a synthetic steroid, is an anesthetic. Steroid hormones affect all biomolecular activity in the body, but they act slowly as they interact with the receptors in the cytoplasm of the cell and plays a role in gene expression. The inhibitory effect of alfaxalone, the anesthetic, acts rapidly on the brain. It was proved in 1984 that alfaxalone activates GABA-A receptors, which explain its rapid onset. It was also found out that all enzymatic biolab activity needed to synthesize natural steroid exists actually in the brain and its peripheral glands, specific adrenal or ovarian, etc. So biosynthetic steroids can be synthesized in the brain. These are what have become labeled as neurosteroids, and that's why the author says the brain on steroids. Some of the metabolites, the biosynthetic steroid activate GABA-A receptors using allosteric mechanisms similar to benzodiazepines, mechanisms such as deoxycorticosterone metabolites and progesterone metabolites. At the time of writing this book, the author says a new synthetic neurosteroid is being developed, ganaxolone. I looked this neurosteroid. I looked it up. It is on the market now, approved by FDA for treatment of certain types of seizures, that is a deficiency disorder. Certain type of seizures come because there is a deficiency of certain substances. Neurosteroids are 100 times to 1,000 times more potent in effect than GABA. They are also regularly involved in the synaptic inhibition. They also very likely be involved, can be very likely involved in disorders that affect the brain and mental health. So GABA is known to be instrumental in the development of the brain, since neurosteroids act similar ways to GABA and are markedly more potent. It is suggested that they very likely have the role in the brain development. They're similar to, because they're very important in the synthesis and in the cells. So they start with our development as we're developing. GABA-A receptors are subject to a wide range of drug interaction and has been seen, they also have been found to interact with drugs that are anciently known to mankind, such as a human. They interact with certain drugs that are very ancient and anciently known to humankind, such as alcohol. So alcohol is also a drug. It's a chemical that alters the functioning of the brain. It's a psychotropic. It's psychedelic. It affects the mind, how it works. So alcohol and GABA. So they work now. Now we've seen so many products that affect the mind, act on this very particular substance and its receptors. From the soma, from the anti-epileptics to the anxiolytics to the many other ones. Now, alcohol and GABA. Alcohol, similar to opium, is a very ancient drug and dates back to Sumerian times. Archaeology traced its use to a Neolithic era. Fermentation has been the key to producing alcohol using all kinds of sugars or starch containing plants, grains, and fruits. It has been used in the analgesics and to produce psychological effect and as a disinfectant. So if you've been watching as a kid, or if you are my kind of generation, you've been watching those Western movies when you were a kid, you see that when a guy gets a bullet, they give them something to bite on and they pour the alcohol on the wound and extract it, and that was used as an anesthetic and the same time as a disinfectant. Clinical usage of alcohol became possible with the development of distilled spirits in Southern Europe from grapes, and that was called brandy. Northern Europe mashed grains yielded vodka and whiskey, such as what came from areas such as Russia. The 17th century, Dr. Sylvius, also known as Dr. Franciscus de la Boe, Leiden University, distilled pure malt spirit using juniper berries, initially called Geneva, then Geneva, then the very popular brand gin. The associated drunkenness and other issues had legislators canonized its consumption and placed restrictive laws on it. Clinically, alcohol can be an anticonvulsant, sedative, hypnotic, resembling thus all drugs that affect GABA-A receptors. It causes sedation, loss of coordination, amnesia, and cognitive impairments when it reaches 20 to 50 millimoles in the blood. It reaches certain level of the blood, it causes these side effects. When it reaches blood levels such as 100 millimoles, it could be fatal, or it is fatal. GABA-A receptor sites are very important, interact with a variety of drugs, highly expressing the thalamocortical neurons that are believed to be responsible for sleep and wakefulness. Hence, like you see, most of the drugs that act on them also act as hypnotics, because they regulate most of the action. They are on those tracts of in the brain that is responsible for wakefulness and sleep. So, as we have seen, most drugs and substances that affect GABA, the neurotransmitter GABA, and GABA receptors has an effect or produces these things. Sedation, they're also hypnotics, they act as hypnotics, and they act as relaxants, they act as anti-epileptics, and they also cause dependency. So these particular neurons, this particular neuron and its receptors in the brain, which is really widely disseminated in the brain, have those effects. And if you remember when we studied when we talked about the other medications that act on as antidepressant and as antipsychotics, they act on different receptors and different neurotransmitters. Now, I think we can get the idea on what work on what and how and why. We also, the book also, from the book that we are reading, we found out that these receptors and the medication that affect them, we have also in our body substances that work on these receptors, that's why they are there, and that affects them. That's similar to the medication we have, but acts in a different way, because some of them are rapidly recycled. That's why, for example, we do not get addicted to our own substances we produce, because it gets metabolized and gotten rid of easily. But the other substances we introduce to our body have different effects and can stay longer in the body. And if they accumulate, they affect the body and the brain in a certain way. So this was for chapter 6. Let's listen to some more Divertimento. This is actually the symphony. Okay. Okay. And the next chapter, chapter 6, is going to be very interesting, or similar interesting. It's going to be, we're going to go through the chapter that talks about cannabis, mostly about cannabis. I'll start and talk about the first part will be about the history of cannabis and about the people who were against cannabis, and we're going to talk about eugenics and about supremacists and about Hallie Aslinger. I personally haven't grown up in the US, but I've been here for long, and my parents were not here or did not grow up here. So I don't know anything about those facts, but I trust the people who present this, and especially when you have a very respectful institution that presents such books, that they did their quiet diligence and uh, uh, while researching facts. So, and besides, it's in the libraries, and so I don't want. I'm not in for defaming anybody or attacking anybody. I'm just presenting what the book is presenting. But my interest is actually in psychotropics and mental health and the importance of mental health for the overall health of society. Well, thank you, and then let's see you next time. And I hope I'll see you subscribing and sharing a comment, inviting me to subscribe and share uh, what you what you are presenting to the people and to the people in this community on YouTube. Okay? Well, have a nice night and stay warm and hope you the best.